Welcome to Data and Biotech, a podcast from Cornell where we explore how companies leverage data to drive innovation in life sciences. Every 2 weeks, we sit down with an expert from the world of biotechnology to understand how they're using data science to solve technical challenges, streamline operations, and further innovation in their business. Here we go.
Kevin Brown, welcome to the Data and Biotech podcast.
Thanks, Ross. Great to be here.
Awesome. Well, just to kick us off, would you mind giving us an introduction to your background and what led you to Standard Model Bio?
Yeah. So my background is originally in pure math. And then I realized I did not want to be a mathematician, just for a lot of reasons, although it was quite beautiful. So I joined a brain-computer interface lab in college. That was absolutely awesome and very cool. I went to grad school for that and got to work on early versions of what we would call Neuralink today, invasive neural implants to control robotic arms or virtual versions of those. And while I was there, we had to segment some gray and white matter in some brains to prepare for surgeries. And deep learning was just taking off, and man, it worked, right? And so it was just one of these, oh man, the world is different now, kind of moments when I saw the first few results, and I thought this is what I should be spending the rest of my life doing. And some people have described that in neuroscience the first time they heard, neural recordings. For me, it was the first time seeing segmentations that just worked. So I joined Siemens Healthineers to work on computer-aided diagnosis, which was absolutely awesome. And that led me all the way to foundation models in ways I think we can unpack.
Was there a specific, GPT-3 moment where you realized that foundation models were going to be, sort of inevitable in biology? Can you walk us through your thought process there?
It was really important to record not just from neural recordings, but neural recordings all over the brain. And not just cells like neurons firing, but also diffusion-weighted imaging, structural MRI, optogenetic stimulation of the brain. All of those things kept adding more and more information, the more modalities and other sort of sensing ways about a system that you could engineer and put together at the same time. While I was, doing this brain segmentation, I realized also, oh man, like deep learning is going to render some of the interpretable approaches we'd been taking a little bit less performant. And so when I had that oh man moment, and I joined Siemens Healthineers, I left grad school to join them because I was like, this is the future. When I was at Siemens, we had scale and we started to notice that, look, it's less about particularly thinking what are these particular layers that we need? Should we use this activation function or that one? That period was rapidly ending. And we realized, oh man, we're building systems that can be used millions of times a year. We're sitting on a lot of data. What can we do with that? And maybe we should just scale data. Like maybe that's the simplest thing to do. So we did that. We built a federated learning system that, to my knowledge, is still running, which is awesome. That was for computer-aided diagnosis for lung cancer. But I remember looking at a lung nodule, a CT scan for lung cancer diagnosis. I'm thinking, man, it could be really cool if we could combine genomic assays or pathology information or longitudinal electronic health records. All these are relevant and may even, bear down on whether or not you expect that to actually be a lung nodule or a false positive or something like that, or indicate specific treatment regimes that would work for a patient or something like that. And the response was, well, look, we make tools for scanners and for radiologists primarily in that group, which is an incredibly noble thing. It drives down the cost of healthcare, it increases quality, it lets it spread globally in ways that are otherwise difficult to do given the labor shortage of radiologists and the technical expertise required. That's a very awesome thing, but I really wanted to do this multimodal thing. So I joined Bristol Myers Squibb, where they had, one of the world's richest oncology data sets, rigorously recorded at the cost of billions of dollars through many pivotal, really amazing clinical trials. And that was an absolutely awesome experience, partly because, in pharma, the domain expertise is just higher than almost anywhere else in the world. If you want to talk to a world expert in cell therapy or gene therapy, like you put time on their calendar, you call them up and then you just ask questions and they're happy to talk. You want to talk to someone who really understands how imaging works in clinical trials or should work or could work in the future. You talk to those folks. Likewise for biostatisticians. I was really able to just some of the most productive knowledge immersed times of my entire life, including undergrad and grad school, were just being able to walk the halls of BMS. There was a moment, a GPT-3 moment, which really was literally GPT-3 moment, where I read the GPT-3 paper. And, we've been building data science models in drug development, right? So human data predicting outcomes of clinical trials, let's say, or response to therapy for a patient or something like that. Those were great, but I went, oh man, there's this one figure in the GPT-3 paper in particular where it shows the performance on several downstream tasks, like 100. And some of them you go from like 0% accuracy all the way to like 100 at different scales. Like sometimes it takes 6 billion parameters, sometimes 13, sometimes more. And I thought, man, that's going to happen in biology. And we're going to make these multimodal. It's all going to go into one giant foundation model. And we're going to figure out a way to make it work. I don't know exactly how it's going to look, but that's what's going to happen. That was sort of the GPT-3 moment. And then the rest of, the next couple of years, we're figuring out where and how to do that.
Yeah, awesome. So with that background, can you give us an introduction to standard model bio and maybe how you think differently about foundation models for biology?
Yeah, for sure. So I realized we needed, multiple modalities, and that goes back to my time in grad school. But then I also realized we needed data scale. And there was a third element that really came down to me, that I started to understand when I was at BMS, which was that domain expertise, that last like 10% was critically important, right? And it's not something that every like every foundation model can do all of. Like you can't own predicting outcomes in metastatic melanoma and non-small cell lung cancer and early breast cancer and cardiovascular disease if a patient has a previous history of a GLP-1 or a diabetic history or something like that. All of those details are critically important, and no one can build a model that does everything, but maybe you can build one that facilitates any downstream task. So I thought, where would be the best place to do this? And where would I get the data to do it? So I thought, well, look, maybe I do this at BMS. BMS is a fantastic place with incredible data, but that data, like every big pharma's data, is highly biased, and it should be. Right? It's mostly your drugs, mostly early trials, mostly failed trials, and older standard of care. Not a lot of your competitors' drugs, and that limits the generalizability of any foundation model that you want to be very general. So I thought, okay, it needs to be more than just these vertically specific things.
Yeah, awesome. So, in terms of, how you think about foundation model that facilitates downstream tasks and enables a variety of different use cases, one of the things I've heard you argue is that the patient is not a document. So can you, unpack that and sort of like give us an understanding of like how you think about the patient mathematically and, why is, thinking about a patient as sort of like a bundle of text, fundamentally limiting.
When we first started this, we thought, look, we're going to use this with text, right? We thought we'd use a large language model backbone. You'd be sort of silly to not use an LLM at all, right, for biomedical AI, because they've already been trained on like all of PubMed, right, and most of the scientifically published literature, for better or worse, and that makes them tremendously powerful tools. And so we thought, well, look, language is a very general space, right? It's almost by construction designed to describe anything. So maybe we should just map everything into it, whether it's radiology images through radiology reports, whether it's pathology images through digital pathology reports, whether it's any other molecular report. That was fairly fruitful. It's not that difficult to do. To give you an example, we used an existing pathology foundation model published by Bioptimus. We took that, we combined it with a Llama 8B model, and in 2 hours, you can get state of the art for medical image understanding, at the time on like a single H100, including debugging, right? So we were thinking, oh man, this is the future. But then we realized that when we wanted to incorporate other modalities that weren't as readily mapped to text, we were running into some walls. So my favorite example of this is whole genome sequencing, right? You've got, 3.6 billion base pairs, and you're trying to compress that into text, but most of the text you have that's linked for any one of those things is like KRAS positive, EGFR positive, like great, right? ROS1, okay. But that doesn't really give you a rich enough way. So if you take some of these contrastive approach or the traditional vision language or other modality language models, it wasn't really going to work to then feed it into like a language aligned embedding to add to the context to the LLM. So we switched a little bit to a JEPA style model. And that was one of the first things that like really felt right. Like we think, oh man, like we are really respecting the complexity of what it means to be a human patient. Right. Every physician, every clinician knows that a patient is not just what's written down in the notes or even just communicated around. There's other subtle things. There's, oh, well, we assumed X, Y, and Z because of their previous history or things that don't get written down, or maybe they just get said out loud separately. There are visual things, right, that an epileptologist can see when they look at EEG traces, right? And they don't always, again, make it into the report. And so we thought, wow, this works really well for radiology and pathology, but for other things, it doesn't. Let's map it into a shared latent space. So we still use language models. Those language models can interpret physician notes or electronic health records, map them into language space, and then that language gets projected into this shared embedding space. Likewise for images, likewise for digital pathology. And this actually turns out to be not that difficult because every single AI model, more or less, maps input data to some compressed representation that's a vector. And you just need to find a way to take that vector and map it onto the shared internal vector or whatever. That really made us happy. We thought, well, what's the best way to pre-train this, right? How do we make sure that we're getting a fairly good model? Should we use, data corruption techniques like mass language modeling, for instance, or mass image modeling? And we really were inspired by some of Jan LeCun's work with the Java models, joint embedding predictive architecture. One of the things we loved about that is it naturally fit into this patient is not a document perspective, where we take a lot of modalities, imaging, pathology, EKGs, we map them into a shared space, and then you predict what that space should look like at time t+1, and then t+2, and then t+3. And that shows it's almost by definition a trajectory of a patient in this abstract space of patient space. It's defined by the different measurements that you could make about that patient. And it's never limited to the ones maybe you have for that patient. If you have an EKG, great, it can shine a little more light on where they are in that space. If you don't, that's fine. You kind of marginalize over it. And we felt like this was the first thing that really respected sort of patients, and also, let us think about patients as moving temporally. They're not just a snapshot of the document you have at a particular time. It's not just about predicting the next word you'd see in a note or answering a question that may go into a radiology report. It was really about, hey, here's the patient today. Where do we expect them to be?
What I'd love to do is just kind of say back to you my understanding of the way the model works, and like it maybe like work with you to define some terms just to make sure that that everyone's coming along with us. So first of all, like ultimately what we're trying to do is create an embedding for each patient based on their medical history up to that point, that captures all of the information from all of the different modalities, all of the different ways that we measure that patient's health throughout their health journey, from everything from CT scans to blood work to genomic profile, clinical notes. All of it ends up in this single vector, the single list of numbers that says, this is who this patient is at this given point in time. And the JEPA approach that you're talking about, my understanding is that, rather than trying to take an approach where it's like we're like trying to make the patient noisier and have the model predict who that patient is, what you're doing is trying to predict what that embedding will be at some point in the future based on what's happened previously. So it's like you're like stepping through time and saying, in 2020, this is how we thought the patient would look in 2021. In 2021, this is how we thought the patient would look in 2022, et cetera, up to the current moment, where you can then take that embedding and you can do things like predict what that patient might look like in 2027 based on their medical trajectory to date. Am I thinking about that right? Or would like where
Yeah, exactly. You're thinking about it right. And I think one of the things that it affords, like once you get into this regime, one, you can build anything off of these embeddings that you want. Just like every AI model takes data it's seen and produces a compressed representation and then does something with it, whether it's a classifier head, basically putting logistic regression, or, a softmax head on top of a classifier, whether it's regression to a single number or several, or whether it's a diffusion head for producing an image, for instance, in like a stable diffusion style model. We know that that's really, like almost like Play-Doh that anyone can go build things off of, which is really cool. But the temporal nature of it, what gets us excited is, you know how like in physics, sometimes working in in the space of differential equations, it makes things easier, right? When you think about the equations of motion, right, it's easier to specify them in terms of change over time and space, rather than mapping out the explicit trajectory, right? Integration is kind of hard, but differentiation is kind of easy, and it's easy to say, hey, look, if I perturb this a little bit this way, this is where I expect the ball to roll or the planets to move. And I think where we're moving, like, I really like this sort of physics, very loose analogy, right? But when you think about the patient, like I feel a little bit sometimes like, Kepler looking at the stars and trying to figure out the equations of motion, right? And writing down like in a notebook, oh, here the stars were in this position, that position, this position, and then trying to figure it out. And then once it like sort of clicks that, oh, wow, you can describe these things in, these very elegant mathematical ways in terms of like how you expect them to vary over time, everything kind of makes sense, right? And going through classical Newtonian mechanics. And we thought, man, like, I want to get to that place for a patient where we say, hey, look, this is where you are, this is where you're going. And then you can think like, you can start doing these counterfactual games, right? With the appropriate statistical rigor. Like that was so drilled into me at BMS, but like I came out with massive respect for the biostatisticians there. And you think about like the difficulties of really designing systems that ascertain what's true. But I love this idea of counterfactual reasoning of like, hey, look, this is the trajectory you're on. And if we do this intervention, whether it's something like exercise, whether it's a GLP-1, whether it's a cancer treatment, we expect you to maybe go over here, right? And then you can say, hey, look, not only did I do counterfactual reasoning, or I jittered some things to see how likely it was that I was going to go each time with sort of some Monte Carlo sampling or something like that. You can say, hey, look, am I on the right track? I did this intervention. That means I expect that I'm going to be over here. Am I actually over there? Is this therapy working? And I love this idea of thinking about the good spaces of patient space, right? Where you're have longevity expected and things like that. And these sort of like bad attractors, right? Like serious adverse events, right? Or cardiovascular events, or like a heart attack, something like that, or like metastatic disease, and trying to push a system away from that. Right. And you can sort of start thinking, hey, maybe, these therapies are basically bumps, and maybe they work for a period of time, like a targeted therapy that works great in lung cancer for a few years before you develop resistance or something like that. And I think this is just a way of thinking about it that is just kind of elegant, and also, respects the fact that patients, like, they change over time. They have many measurements that matter. They're not just what's written down in one specific document. They're not an ICD-10 code.
I want to spend some time on the modalities aspect of this, and then kind of return to the temporal nature of it, and like how you think about that, those counterfactuals, and like how a patient moves through time. My understanding is that you're taking these very different data types, from radiology images to clinical notes, and you're sort of treating them differently within the model architecture, and you're utilizing different assumptions in the way that you process each modality in order to get the most information from that particular modality as it's being brought in. And that also, there's a way of handling this that means that you don't have to have every modality for every patient in order to effectively model that patient's trajectory. So can you maybe help me understand, like, how those different modalities are treated?
Yeah, so each modality gets its own specific encoder to begin with, right? So EHRs can be ingested by language models fairly readily, but then likewise, let's say a radiology image, right? We use vision transformers in 3D. It takes really about 3 lines of code to build them today, right? It's a fairly straightforward thing. Of course, then when you want to do a little more fancy things, you have to think about various data augmentation techniques and things like that. We've, for instance, trained on 1 million CTs, maybe 1.5 million now, but 1 million oncology related ones. And that does allow us to sort of encode various kinds of CTs and then put them, into that same unified space. Will be coming public with that soon. But like the idea being like we treat vision, computer vision, in a way that we know there's good standards for how to treat it. We don't have to reinvent the wheel there. And if there's a better radiology model, we can also use that too. Likewise, digital pathology. We use other digital pathology models right now. I think the field is like a wash in them. I think it's a really intense and crazy space where there's like all this churn. And, 1 week you see this $50 million whatever model. And then in the next week, you see someone train a very similar one for a few thousand. And we thought, what, we're going to like just sort of use what other people have produced at this point. And that's totally fine. Right. So you can ingest, using best practices, digital pathology data with all the careful thought that has to go into that, and then map that into that shared latent space. Likewise with an EKG, likewise with, it could be EEGs, it could be brain MRIs, it could be, chest X-rays, things like that. The important thing is we don't have to develop every single encoder ourselves, because I think that's too big of a bite to chew. But they do that in imaging. We do that in genomics. We are doing that soon in multiomics and other things. And there's a number of reasons for that. The other thing that I love about it is that if you're missing a modality, that's okay, right? And this naturally pairs with the way the clinicians have to operate. Sometimes you don't have a test that you would like to do for a variety of reasons, or it's missing, or it didn't come at the right time, or maybe the endoscopy really didn't get what you wanted to see, and you have to think about how that information would get built in otherwise, things like that. That means that you're not totally bound to these very low scale regimes where you only have really highly paired data for every single sample. I think that's unrealistic, and you're not going to get the scale that you need to build the size models that you want to really reason about these things. And I want to maybe spend a little bit of time. Why do we care so much about so many different modalities? Right? What if you just did a vertical model for, okay, here's my radiology model, here's my pathology model, here's my EKG model, here's my genomics model, and then I'll just sort of integrate them later. I think that doesn't work for a number of reasons. One, because even at a low level, you get some interesting cross attention, like cross, feedback across these different things. But two, clinicians know that like they don't even operate in these intense verticals. There are some silos, but even like a radiologist doesn't just produce a report and that's the end of it. Oftentimes, like at a serious, hospital, there will be feedback. You can call them up, you can ask them for clarification, you can double down, you can say, hey, well, the patient was reporting this other thing, can you take a look? There is that kind of crosstalk, and so it makes sense to build models that also have that communication built in. And so I think that is a future. One of my favorite examples about this is cardiotoxicity, right? To show just how broad these things can be. Cardiotoxin in oncology. So in radiation therapy, you can have adverse events from radiation therapy, especially in the cardiac sense. And you can triage patients according to how likely you think it's going to be that they're going to have that kind of event. So you can look at pericardial effusion in a CT scan. You can combine that with routine, electrocardiograms. You can also combine that with the longitudinal EHR that would obviously show different kinds of risk factors, whether it's as simple as body mass index or similar, previous obesity history, smoking status, things like that. Well, all of those are going to be relevant. A clinician's going to compare all of those things, and why not do it in sort of raw signal space if you're building a model? It also shows how interdisciplinary we know that this model has to be. So the verticals can't even be like oncology, right? Because even in oncology, now you're in cardio, right? And those cardio models are going to get better even if they're just trained on general cardio patients. And likewise, you're also going to be in immunology, not only because of immunotherapies and the side effects that you have there, but like, look, there's gastroenterologists that only see oncology patients. There are cardiologists that only see oncology patients, and it's only going to get more complex. And so we didn't really see there be, like a limiting factor there. And then once you're that wide in the horizontal, like, man, there's no way you're going to own all those application layers. Like, you cannot credibly claim that you are an expert in cardiotoxicity for early lung cancer and the progression of metastatic disease, in melanoma. Oh, and by the way, just general cardiology, like that's insane. But you can do that first 90% is very similar.
Yeah, that makes a lot of sense. And also, like the goal is a foundation model that like is the best, most complete representation of the patient that's possible. And there's information in all of the different modalities that can inform that representation of the patient through time. And, so to ignore that is to, fundamentally, restrict yourself from getting that full representation of the patient. Another question I had about the modalities is like, how do you prevent a high signal modality like imaging from, dominating like, noisier modalities like genomics?
That's a great point. And this actually plays into the temporal aspect. And this is a problem we're actively working on. But if you're an oncology patient, you're going to get several CT scans most likely, right? And they're going to be at regular enough intervals. And so CT is a great place to go. That's the other modality that we went into besides longitudinal EHRs, because the most regularly paired. And then at baseline, you may have sequencing done, right? But it's not going to be like all the time, right? Likewise, a resection with pathology, very important to baseline. It's not going to happen at the same frequency as a CT scan. So there are ways in which you can get better clarity of where a patient's going on the trajectory, and then that can be updated with seeing what you expect to see and the MRI. The other thing that we do, which I think is important, is you have to kind of ground the model a little bit. So you need to either be able to like, reconstruct the signal you would expect to see, right? Like, hey, am I seeing a plausible MRI at this point in time for where I am in embedding space? Am I seeing what would be a plausible Dr. Note or ICD-10 code at that point in time? Or am I seeing an EKG that looks like I expect? That, I think, is a pretty important thing to make sure that the embedding space stays relevant and it doesn't collapse into sort of repeating sort of trivial embeddings. And that was a major unlock for us.
Right. So if I'm understanding it, what you're describing is like that you're using like a joint objective when you're training the model. You're using like the supervised version of the model where you're trying to predict the next quote unquote token, the next thing that would be in that patient's medical record, and then you're also using JEPA as you described earlier.
Yeah. That's right. And we were pretty happy, in comparing this to just, let's say, predicting next token or medical history event. We worked with Memorial Sloan Kettering with an absolutely amazing, data set that combined things from digital pathology, imaging, longitudinal EHR, and I would say EHR plus, because it, contained a lot more than you would expect, and very temporally done and just very rigorously curated. And that was an amazing collaboration because that allowed us to test this out, right? And so for model size, even for bigger models, compared to bigger models, it does provide a lift. And interestingly, the bigger lift is not in some of the simple things, but it is in producing like certain kinds of toxicity or progression to specific organ systems. And also it works better in more complex diseases like sarcomas. And I thought that was like pretty cool because I think there's certain things where you're maxed out. You're not going to get too much more prediction in terms of like predicting outcomes if you already have maxed out sort of what that data is capable of doing. So we were really excited about that. Like that when those results came back, I still remember Urshad, who really led that work. He's fantastic. I think he did a lot of it probably while he's still defending his PhD. But I was in India at the time visiting for a friend's wedding, and he called me. And I immediately got out of bed, and I knew he was like dancing on the other side of the world, and we were both sort of like jumping up and down, just like, yeah, this is cool. And it really felt right.
There are a variety of reasons that makes sense to me as well, because, I still sometimes can't get my mind around is the idea that the model is constructing the target embedding as it's training the predicted embedding. And so like having that supervised objective, like to me, at least makes sense in terms of like helping the target embedding along toward something that the predicted embedding is getting there when it's, using that to calculate the loss inside of Jeba.
And what I love also about that I think makes it possible to do is like, look, if I take this additional measurement, am I going to meaningfully change where that embedding would be or what something else would look like in the future? Is it going to inform what I would see with other modalities? And then that lets you optimize the kinds of tests that you would run, right? Whether that's in clinical trial development and picking the biomarkers that you would want. You can maybe figure out which ones are redundant because they're not changing the expected trajectory of a patient. So don't do a measurement, or whether it's in a system that's eventually clinically facing. And that makes me pretty excited because everyone likes to say the right drug for the right patient at the right time, which I think is cool, but it's also like the right measurement for the right patient at the right time, I think informs all of those decisions.
So this was another question that I had about the modalities is one of the downstream tasks like figuring out what the information gain would be hypothetically from taking a particular test from a particular modality for a particular patient in order to like improve the embeddings?
Yes. Yeah. We would love to do that. And like, there's all these like nuances for doing it right. So we can like play around with it and we can do these basic assessments. And we do. We are actively looking for like really awesome biostats collaborators that would love to just take it and do it. I'd also mention like in terms of these downstream tests, like right now, all of the models are open source with very permissive commercial licenses, right? Like you can go and download them and you can use them and you can do various things with them. So like, we really want everyone to go take it and do beautiful validations with it and beautiful downstream tasks, right? Like, and we'll even help people do it. We worked with one group and, the paper's not public yet, but it took them like a couple hours to get a positive result. And then, they submitted the paper like a month or two later, which is fantastic. Likewise, we've repeated that with some other groups. The other day, one of the other like really highlights for me was I just told Claude Code, hey, like go download the standard model and apply it to this data set from this paper. I didn't provide any links, right? And it like searched the docs, pulled it down, and didn't require any intervention. And I was like, okay, that's cool, right? I think this is now I feel like we're really speeding up biomedical AI in like a meaningful way. That made me happy, yeah.
The tools that are available to fine tune fine tune foundation models across tasks have just improved dramatically, and that what that ends up doing is just expanding the potential list of end users who could think of a problem that a time aware patient embedding could help to inform. I'd love to get to some of those to some of the tasks that you think would be most appropriate later, but I think one of the things that I'd love to understand a little bit beforehand, when you think about the temporal nature of the model, how do you separate something like disease progression, like the model is picking up on this patient is likely to, have some progression in some disease that they're experiencing, versus there's some treatment effect that happened earlier that is then altering the patient's trajectory. I was just having trouble thinking of like, if the tumor is shrinking after chemo, is the model learning like chemo works, or is the model learning like this type of tumor was regressing, or like how do you think about like counterfactuals and that kind of?
We think deeply enough about that to know that we should have other people think even more deeply. And part of that is, look, like causal data analysis is really hard, and understanding the heterogeneous, treatment effect, like a treatment effect for a specific patient, is really difficult because, you can't go back and give the same patient a different treatment and see which one worked better. You can't go back in time. So that makes it really different. And there are very good statistical techniques to do that. They do require a lot of nuance. I think it does permit that kind of analysis, but you have to be like, going back to the BMS days, like you got to be really careful, or you will lead yourself down this road that is just like not statistically sound.
Another thing about the time aspect of it, like we talk about time in terms of like t-1, t and t+1, but sometimes you're trying to predict what the patient's going to look like in 2 years, and sometimes you're trying to predict what the patient's going to look like in 6 months. For people who are sort of like downstream users of a model like this that are trying to predict the next thing that's happening, how do you advise people to think about like time in the context of the model?
Yeah, that's a great point. So we would love to make it very time agnostic from very small scales to very high scales. Where I would love to see it go is, hey, look, in the next hour, what should I do with this patient? All the way to like in the next year, what should I do with this patient? Or people build systems that do that. I do think that will be difficult. Like we're not in the like real time sense yet of like, hey, you should do this test. But like there are scenarios where you would want to be able to make those decisions, right? With a patient that's like likely to go into status and that's going to change like different treatment things that you could do, or has a sepsis likelihood or something like that. But it's like I think one place it could be cool, like applied in a cool way. And there was some actually like LLM based work at NYU Langone that did a lot of this, which I thought was really awesome. The senior PIs on that work, Chang Cho and Eric Orman, there, absolutely fantastic researchers. They did implement in real time being able to predict things like mortality in prospective shadow mode, which I thought was great. I would love to see it used to like reduce alarm fatigue or something like that, right? Like, hey, look, like, what is the most important thing to alert a clinical team to for this patient at this point in time? Or what is the test I need to do to avoid those things? And how can I queue up the recs for the things that will need to be done? We're not there yet, but I would love to see that, like, because you're right, like patient relevant time scales can go from seconds, right, in a stroke to like years in terms of like a exercise intervention.
So even though you can't know what the time scale is that you're operating on, it does allow you to do things like predict t+1, see what the embedding looks like, and predict t+2, predict t+3, predict t+4, and see like in the absence of intervention, this is what the patient's trajectory looks like through amorphous time, and then also sort of inject a study or an intervention in the middle of it and see how that influences sort of the trajectory of that patient. Am I
Yeah.
thinking about
Yeah,
that
yeah,
right?
yeah, that's right. Yeah.
Let's go back to foundation models versus vertical models for a minute. So like I know that like you talked a little bit earlier about how medicine is not naturally narrow to these disciplines, that like there's information from each discipline and each modality within each discipline that sort of feeds into the overall picture of the patient. Can you just talk about, do you have any, like, what evidence out there supports the claim that, like, narrow models are more brittle, or that the approach that you're taking for these broader foundation models is, definitely the right way to do it?
So formal evidence, like I wouldn't go so far as to say that. Like it's not like we've done a register, a register study for it. But a lot of it is just sort of reasoning through it from first principles. Like I think that we know that multiple modalities are relevant for patients, or we wouldn't have tests from those modalities. I think the question is, what is the best point to integrate them? And the way they're integrated right now is largely text and then sometimes follow up text, right? A radiology report that gets read, a few words that describe a genomic assay, right? EGFR positive or whatever. Those are very valuable today, but integrating them at the end at the text is like, by the time you get something all the way down to text, you've reduced the content. Like even in a normal LLM, right? Like predicting the next token, one token has less information than the embedding that you use to predict that token. And so it makes sense to communicate across these modalities and embeddings in one way, shape, or form because there's just so much more richness there. You can only ever destroy information by converting it into text, even if it makes it more legible for a human. And then the question is, where should that communication happen? Should you do it all the way at the end, like with the vector that you would use to predict the next token? Should you do it earlier? If you do it earlier, do you get benefits? And that's what hasn't really been rigorously assessed yet. I mean, I know there's some papers and research that looks at these levels of fusion, but I think some of it hasn't really explored the scale at which you really could or should be doing these things. And so I think that, it's going to make sense to have multimodal models. Now, medicine does get siloed, right? There are tracks, obviously, and there need to be for reasons of education. But I do think in the future, and I don't know when, but I do think that some of the communication that happens right now by text, either by physicians or by agents, right? Or like you could have a radiology to text report generator, and those reports then get fed either to another human or an agent that is interpreting all of that text and combining it with text from the genomic assay. I think in the future, it's not going to be an agent or a human at the top just pulling the text. I think the communication is going to happen implicitly at a lower level from modality to modality, from a model that is sort of naturally communicating because there are embeddings that are talking to each other, from radiology to pathology to genomics to general patient information. And the human will definitely always be in the loop, but I just think that the communication can happen at a lower level because text is just such a lossy medium, right?
Right. So the argument here is basically that every small piece of information that's gathered throughout the patient journey should be treated as like gold, as like really valuable, and that this embedding space that you're constructing across modalities is designed to capture as much of that information as possible and not lose any of it through the ways that humans communicate with each other inside of hospitals, inside of EHR systems, and that the EHR systems could potentially benefit from having systems that reference that, condensed representation of all that information. Am I
Yeah, no, that's right. Because and this isn't, really like new when you talk to like a radiologist, like they know there are things about that scan that they're looking at that don't make it into the radiology report for a number of reasons, right? And like it's there, they can see it, right? They can look at a tumor margin and maybe think, that doesn't look necessarily as good as I would want, but like how do you quantify looks icky, right? Like, you know what I mean? But it's there. And likewise with, digital pathology, sometimes it's not just about counting up, like a PDL1 measurement. It's a little bit more nuanced, and some of that nuance doesn't get put in for a number of reasons, but it is there and it can be picked up. And I think that the labels for it are difficult, but if you have a self-supervised task, which is like predict where this patient's going, that's highly patient relevant, so it's meaningful, and it's also, I think, naturally pulls out those things that matter for that patient, even if they don't get written down explicitly.
Yeah, I think that makes a lot of sense, and also it highlights for me the way that uh, our healthcare system is sort of like trying to get everything into discrete space. Everything gets a code, everything gets a category. It's a binary, like you're either diagnosed with the thing or you're not diagnosed with the thing, but that the embedding representation can capture like a more continuous journey of, what those different radiology images look like, that say, we're moving in the direction of a diagnosis that looks like this, even if it is not diagnosable at time t minus 3.
Yeah, yeah, that's right. I completely agree.
I want to talk a little bit about the training data that goes into this, and also how that plays into, like, if a researcher is listening who is sitting on a bunch of patient data, how does the way that the data is prepared to go into this model relate to the way that somebody who has a lot of patient data and wants to get these patient embedding representations out, how would they interact with the model in order to do that?
Yeah, that's a great question. So it takes way less data normalization or curation than it used to, right? In fact, when we're faced with the table, we back it out into text anyway because it's easier for the LLM to ingest. I think so. Don't be scared away by thinking, oh no, like I'm gonna need to like spend hours and hours and hours like making sure all of my columns line up to whatever I think that the columns were that the standard model was using. I don't think that's maybe the best way to think about it because it's going to go back into text anyway. I think anytime you have those raw modalities, it's fine. There's relatively limited amount of pre-processing you need to do. And I think that makes it really good as long as the data is accurate and high fidelity. And that part, of course, still needs to be done because sometimes things get mislabeled. Sometimes you're going to accidentally, we've seen people accidentally write micrograms instead of milligrams. And those are obvious to correct when you see them, but like the model may not know that. So those, need to be reviewed. But broadly speaking, it's not that difficult. And we're happy to help anyone do it. But, it's not crazy.
And is there any concern either on your part, or do you think there should be concern on the part of potential users of these models? The data that you've trained the model on is from a fundamentally different distribution than the data they might be feeding into it, like some data shift happening there.
That is a fantastic concern, right? So right now we are oncology forward, although we're moving rapidly into immunology and cardiology as well. That being said, yeah, if you went and downloaded this and said, "Hey, I'm going to use this model that is really good at oncology to predict, neurodegenerative outcomes in my Alzheimer's disease trial," well, you're going to have some issues without like fine tuning that model on some Alzheimer's data, right? I think that would be one thing. I think it goes to a broader point, right? Any foundation model is a compressed representation of the data it was trained on. Right, like that's kind of by definition, and so we want to be sort of this latent, super, like pseudo data layer, so that you don't have to go out and buy all of that data necessarily, right? In order to train your model, you don't need to go buy thousands and thousands of CT scans or millions or whatever. And I think that makes it exciting or easy for people to get up and use. But if you're trying to use that model, no matter how good the model is, if it wasn't trained on cardio data, it's not going to be good at cardio.
If it's a task that is not in distribution for it, or it, for sort of the data that you originally brought in when you trained it, you should be thinking about how can I fine tune this model and then evaluate it. Which brings me to the question of what are some of the ways that you make that you evaluate the quality of the patient embeddings that you're producing, and how do you think about interpreting how good the model is at different downstream tasks that it might be asked to do?
That's a that's one of the core questions, which I think actually doesn't get answered enough, right? So we're at this period where everyone is like, "Oh, I want to build a foundation model," and so they train a model, and sometimes it's only evaluated on one thing or two things, and that's not really what you would necessarily want or need. You really need to evaluate it on lots of things. But the problem and the reason why people don't do that, it's not that they're lazy; it's that they don't have the data. Right? And so you need to go to other institutions and say, "Hey, look, like, can you evaluate this on your use case, on your patient population?" I think taking one step back, if I'm a patient and I want a model that's powering some AI that's making decisions for me, what makes it good? Right? Well, okay, we say the embeddings that the application layer is using are good, fine. But how do we know that? It doesn't really matter to a patient how well it works on average. It matters how well it works for patients like them, right? And this is a fundamental problem. Everyone has this problem. It's not like new, right? It's when you go do a clinical trial, you have a particular population, but that may not be what it's like to be applied for a lifelong smoker who grew up in, I don't know, Greenville, Alabama or New York City, right? And we know that socioeconomics and various demographic things have real effects in patient populations and affect the way that things are applied, whether it's at the drug level or the AI level. We need to evaluate these things very locally. I'm not the only person who said this. I think Nigam Shah at Stanford has been saying things like this. Like local evaluations are good. We shouldn't just have huge benchmarks that are like, hey, this is the lung cancer benchmark. This is the interstitial lung disease benchmark. We need ones that are like really specific to the patient populations that they serve, which is why we're happy to give the model to different people to go evaluate it. Like I actually think that needs to be done more because you see these patients and it's like, well, we trained it on this data. It was really good. We got great results, huge scientific insight, but we really only know if it works at this one institution. Like it needs to be validated on more places, I think.
Yeah. But practically speaking, just based on my understanding, it looks like use the data that you have to get embeddings out of the model, either through inference or through fine tuning, and then inference, and then you use some sort of prediction head on top of those embeddings. It could be as simple as like a regression on the different features in the embeddings to predict a particular outcome, and you say, how predictive is this like linear model or more complicated model of the outcome that you care about. And that's sort of how one would think about evaluating the quality of the underlying embedding representations. Am I thinking about that right?
Is it doing something that's useful? Like in oncology, let's say I'm doing time to event. If I put a Cox proportional hazards head on top of this model that takes these embeddings and spits out, the hazards, can I use that like, to get a higher concordance index or time dependent AUC or whatever the appropriate metric for that particular question is than some other model, right? And that would indicate whether it's useful, right?
Yeah, that makes a lot of sense. That makes a lot of sense. So, from a use case perspective, can you just highlight some of the ways that your open source models have been used in the wild so far?
Yeah, yeah. So one's like, like early onset pancreatic cancer. One's this, thinking about cardiotoxicity. The other is toxicity for general treatment for, whether it's sarcomas to pancreatic cancer to non-small cell lung cancer. Those are the first places where we've seen a lot of the application. Some things that are relevant to clinical development, right? Can you predict when a patient is going to have a line of therapy transfer that would make them eligible for a clinical trial? Can you predict whether or not they're likely to meet the inclusion exclusion criteria for that trial at a specific time in the future rather than just right now, so that eventually HCPs can take the model or applications built on that model and then start those conversations early because you expect the patient to go in a certain trajectory. You can start figuring out what the best trial for them might be, for instance, or eventually, in the like one of my favorite places is thinking about metastatic disease for oncology, and can you predict certain kinds of adverse events? I think it's going to be a huge thing, right? And I think it's going to be fun.
Yeah, the other thing that's occurred to me as we've been talking about the embedding space is just that the embeddings are so flexible in terms of the things that you can do with it. Yes, you can feed it to a deep learning model to predict something. Yes, you can run a regression on them to predict something. Yes, you can do a clustering and segmentation analysis to see who's close in embedding space and who might be like a patient that you think is the right kind of patient profile for a clinical trial. There's just there's a lot of potential downstream use cases for this kind of thing.
That's right. Even for just for communication, right? Like, hey, look, here's a patient that was similar to you. Here's what happened with them, right? I think that could be a powerful thing. Yeah.
Yeah, very interesting. So you've done a lot of work on these models, but you've chosen to open source them. Could you give us some insight into why you decided to go the open source route?
Yeah. For now, honestly, it comes down to validation and benchmarking, right? We need models like these to be broadly benchmarked by not just one academic medical institution, no matter how prestigious, not just 10, right? Like by the end of the year, we want this validated in one way, shape, or form on 100 different academic medical centers. I think that's absolutely doable, but I think it's also really important. It makes it kind of frustrating as a scientist, if I put my scientist, not my CEO hat on, I'm like, well, look, here's this other model. Is it better than ours? Is it not better than ours? Is there a place where there's some overlap? Can we make a comparison? Which one should I use? Well, I can't get their data and I can't get their model. So I don't know, right? The only thing we could do, when faces like you can't control what other people do, you can only control what you do. And what we can do, we can give them our model and we can say, hey, look, at least you're aware of it. You can take it, you can benchmark it, and we will help you. We'll dedicate hours from some of the, finest ML engineers that exist in the world, and we'll help you do that. And that makes me feel pretty good at the end of the day.
What I'm interested in from your perspective, so you already mentioned that you're sort of oncology forward. How would you want people to think about, sort of the limitations of the standard model biomodels that are being released?
Yeah. So right now they're oncology forward. We chose that for one business reason, right? Oncology is, a huge market. But it's also kind of the thin end of the wedge when it comes to precision medicine. You will have people write grants that say, as done in precision oncology, for instance. So we knew that was a good place to start if we wanted to work with, people that have been thinking about this very deeply for many years. And that means that if you're using it and it should be oncology adjacent, it doesn't have to be oncology right now, right? Like in the sense of if you want to predict cardio toxicity, maybe that would make sense, even though it's not necessarily a cardio thing. Or if you wanted to use your own cardio model to predict how things would happen in an oncology space, that could work. We are moving into general chest disease with a collaborator that we'd love to announce soon, which we're really, really, really excited about. And I think that's going to be a pretty powerful thing because it's going to get us into some of the immune-related things, which are also cancer relevant, right? The interstitial lung disease can be a devastating side effect to certain immunotherapies. We are thinking about oncology today. In a couple months, expect it to be a lot more. Yeah.
Yeah. So should I think about this as like you have a model architecture that you think is sort of universally applicable, that models the patient in a really great way, but that your journey is a journey of incorporating more, more patients with different therapeutic issues and potentially which weight differently toward the modalities that you're exposing the model toward based on, what that cohort of patients and their journey through their patient experience. Is that yeah.
And if someone's like, look, I would love to use this, but your model's not working that well on it, like we want to know. And sometimes we use that to identify what data to go after next, right? If someone's like, hey, look, I really wanted to use this on, predicting myocarditis and I can't, well, tell us and we can go try to pump that down.
Well, Kevin, you've been an excellent guest. I really appreciate the time. If listeners want to learn more about your methods or, the work that you're doing or where you're heading from an organizational perspective, where should they start?
They can go to our Substack. That's where we publish a lot of stuff even before it makes it into papers. So blog.standardmodel.bio. That's one of my favorite places. Or just email us. Yeah.
Yeah, highly recommend that Substack. That was how I discovered your work, and really enjoyed having you on to talk about it today. So thank you very much, and look forward to connecting down the line.
All right. Thank you, Ross.
And that's it for this episode of Data in Biotech. If you enjoyed the episode, please subscribe, rate, or leave a review in your podcast platform of choice. See you next time.